My name is Trina Ganguly and the date today is October 26, 2022. Today I'll be discussing my presentation, which is the determination of sex, genes versus hormones. To begin with, sex refers to a set of biological attributes in humans and is known to be the mixing of genomes via meiosis and fusion of gametes. It is a defining biological characteristic and is primarily associated with physical and physiological features, including chromosomes, gene expression, hormone levels and function, and reproductive anatomy. Sex is usually categorized as either female or male, but there is variation in the biological attributes that comprise sex and how those attributes are expressed. One of the major roles of sex is to bring together alleles carried by different individuals, revealing beneficial genetic variance that is otherwise hidden. In addition, there are three types of sex: genotypic, which refers to an individual's two sex chromosomes, which is either an XX chromosome, which is a genotypic female, or an X and a Y chromosome, which is a genotypic male. The next is phenotypic, which refers to an individual's sex as determined by their internal and external genitalia, expression of secondary sex characteristics, and behavior. And last, there is gender. Gender identity entails self-appraisal according to the traits most often associated with one sex or the other, called gender traits, and these can be further influenced to some degree by cultural norms. To add on, humans develop as either male or female depending on the combination of sex chromosomes they inherit from their parents. The human sex chromosomes are called X and Y. And humans who inherit two X chromosomes typically develop as females, while humans with one X and one Y chromosome develop as males. Sex determination is the beginning of the development of many characteristics that influence how a human looks and functions, as well as the societal expectations that other humans have for each other. The process of sex determination begins after fertilization, a process where male and female germ cells fuse to form a zygote, or a single-celled fertilized egg. Germ cells are those that carry genetic information from the parents to offspring during fertilization, and male germ cells are known to be sperm cells, while female germ cells are egg cells. And when the egg and sperm cells fuse, the zygote then divides into multiple cells and later forms an embryo. The embryo includes a combination of a part of each parent's genetic information, including one sex chromosome from each parent. The combination of sex chromosomes that an embryo inherits from germ cells determines what biological sex it will later develop as. A process called meiosis determines the sex chromosomes that the parent's germ cells pass on to their offspring. Meiosis occurs inside the testes and ovaries, the organs, which are the organs in the males and females, respectively, that produce the germ cells. During meiosis, each cell divides twice, which results into 4 cells. And when the germ cells fuse during fertilization, the resulting embryo will have the normal amount of genetic material, including 2 sex chromosomes, because it receives one from each parent. Because females tend to have only X chromosomes, the egg cells that they produce typically carry an X chromosome, while a male sperm cell can carry either an X or a Y. Therefore, the sex chromosome that a male sperm carries determines whether the offspring will develop into a male or female. Sexual differentiation is a developmental process by which genetic sex and gonadal hormones determine the sex of the reproductive tract and various secondary sex characteristics, and influence the development of the brain structure and function, including a variety of behaviors. Sex chromosome genes are the primary factors causing sexual differentiation. In species with heteromorphic sex chromosomes, all sex differences arise from the genetic inequality found in the sex chromosomes of male and female zygotes. In mammals, females have 2 X chromosomes and males have 1, while males also have a Y chromosome, which is lacking in females. The differential representation of X and Y genetic material is the sole source of all subsequent sex differences during development and adulthood, because all other factors such as autosomal genes, cytoplasmic material, and prenatal environment of the zygote are thought to be equivalent on average between both females and males. In addition, some other contributing factors are on-gonadal effects of Y genes. The Y chromosome has cell-autonomous effects outside of the gonads that make Y-bearing cells different from those lacking a Y chromosome. Also, the number of X chromosomes causes differences. In specific, the expression of Xs is female-specific. The presence of 2 or more X chromosomes triggers the expression of the long non-coding RNA excess from one of the two X chromosomes, thereby making all such cells different from those with one X chromosome. Furthermore, higher expression of X escapee genes in than XY cells, since some X genes escape X inactivation and are expressed from both chromosomes in XX cells, so that expression is inherently higher in XX cells than XY cells. To add on, sexual inequality of parental imprints on X chromosome genes, for instance, XX cells have X chromosomes and imprints from both parents, but XY cells receive only the X imprints from the mother, along with some female-specific effects of the inactive X chromosome. And lastly, independent sex biasing factors and their downstream pathways interact, which are summing, amplifying, or counteracting each other, and then producing and reducing emergent sex differences in tissue function. When it comes to genes and sex determination, one gene comes to mind, which is the SRY gene, which is found in the sex-determining region of the Y chromosome. It determines sex in humans, and although many genes are involved, the SRY gene is a key switch, and individuals with SRY develop into males, while those without it develop into females. This mechanism of sex determination requires that SRY is active in half the population, not active in the other half, and this is achieved by its location on a chromosome that is inherited by half the population, and not the other half, which is the Y chromosome. Males produce sperm with an X chromosome or a Y chromosome in equal numbers. Eggs fertilized by Y-bearing sperm develop into males, and those fertilized by an X-bearing sperm develop into females. Thus, the primary biological role of the Y chromosome is sex determination. Also, any mutations in the SRY gene halted the embryonic development of testes, resulting in organisms that possessed a Y chromosome but expressed female phenotypic characteristics. And similar to the SRY gene is the WNT4 gene. The WNT4 gene provides instructions for producing a protein that is important for the formation of the female reproductive system, the kidneys, and several hormone-producing glands. During the development of the female reproductive system, the WNT4 protein regulates the formation of the Mullerian ducts, which are structures in the embryo that develop into the uterus, fallopian tube, cervix, and the upper part of the vagina. This protein is also involved in the development of the ovaries from before birth through adulthood, and is important for development and maintenance of egg cells in the ovaries. In addition, the WNT4 protein regulates the production of male sex hormones. Estrogens are a group of hormones that play an important role in the normal sexual and reproductive development in women. There are also sex hormones. The woman's ovaries make most estrogen hormones, although the adrenal glands and fat cells also make small amounts of the hormones. In addition to regulating the menstrual cycle, estrogen affects the reproductive tract, the urinary tract, the heart, the blood vessels, bones, breast, skin, hair, mucous membranes, pelvic muscles, and the brain. Secondary sexual characteristics such as pubic and armpit hair also start to grow when estrogen levels rise. Many organ systems, including the musculoskeletal, cardiovascular systems, and the brain, are affected by estrogen. There are three main types of estrogen. Estrone, which is the primary form of estrogen that your body makes after menopause. Estradiol, which is the primary form of estrogen in your body during your reproductive years, and is the most potent form of estrogen. And estriol, which is the primary form of estrogen during pregnancy. Furthermore, testosterone is a hormone that your gonads, or sex organs, mainly produce. More specifically, the testicles in people assigned male at birth, and the ovaries in people assigned female at birth produce testosterone. Your adrenal glands also produce the hormone, which is DHEA, which your body transforms into testosterone and estrogen. Testosterone is the main androgen, meaning it stimulates the development of male characteristics. It triggers the development of the male internal and external reproductive organs during fetal development. And natural testosterone is a steroid, in specific an anabolic androgenic steroid. An anabolic refers to muscle building, and androgenic refers to increased male sex characteristics. Also, levels of testosterone are usually highest in the morning and decline during the day, and your hypothalamus and pituitary gland control the amount of testosterone your gonads produce and release. In specific, your hypothalamus releases gonadotropin, releasing hormone, which triggers the pituitary gland to release the luteinizing hormone. And the luteinizing hormone then travels to your gonads and stimulates the production and release of testosterone. And next, progesterone is a steroid hormone belonging to a class of hormones called progestogens. It is secreted by the corpus luteum, a temporary endocrine gland that the female body produces after ovulation during the second half of the menstrual cycle. Progesterone prepares the endometrium for the potential of pregnancy after ovulation. It triggers the lining to thicken to accept a fertilized egg. It also prohibits the muscle contractions in the uterus that would cause the body to reject an egg. If an individual becomes pregnant, progesterone continues to stimulate the body to provide the blood vessels in the endometrium that will then feed the growing fetus. The hormone also prepares the limit of the uterus further so it can accept the fertilized egg. Once the placenta develops, it also begins to secrete progesterone, supporting the corpus luteum. And this causes the levels to remain elevated throughout the pregnancy so the body does not produce more eggs. It also helps prepare the breast for milk production. After the egg is fertilized, it divides over and over until an embryo begins to form. Each step from egg to fully formed embryo is precisely coordinated. A key step in embryo development is making either a male or female reproductive system. At first, rudimentary tissues for both male and female reproductive tracts are present. In males, the rudimentary female tissue disintegrates, and the tissue that will become the adult male reproductive tract develops. In females, the opposite happens, where the male tissue disintegrates and the female tissue develops into the adult female reproductive tract. In addition, male hormones called androgens drive the process that develops male reproductive tract tissue, and it is assumed that female embryos lost the male tissue because they lacked androgen. In addition, two precursor organs exist in the fetus, which are the Wolffian duct, which differentiates into the structures of the male genitals tract, and the Mullerian duct, which is the source of the female reproductive organs. During the third month of the fetal development, the Sertoli cells of the testes of XY fetuses begin to secrete a substance called Mullerian inhibiting hormone. And Sertoli cells are the somatic cells of the testes that are essential for testes formation and spermatogenesis. This causes the Mullerian ducts to atrophy instead of develop into the oviducts, also known as the fallopian tubes and uterus. In addition, the Wolffian ducts are stimulated by testosterone to eventually develop into the spermatic ducts, which are the ductus deferens, ejaculatory ducts, and seminal vesicles. And that is the end of my presentation. Here are my references. Thank you.